Hello, everybody. Very good evening to all you all. Can you hear me? A quick hellos around. Good evening, everyone. Good to see you all here waiting for the session. So, can I have some quick hellos from you all so that I know who all are here? Good evening, Akshata. Good evening, Pooja. Hello, everybody. Hello, everyone, once again. Yes, so welcome to the session, everybody. And let me tell you, especially the ones who are joining the session for the first time. So this will be part C question discussion from Unit 8. And today we are going to talk about the topic microbial genetics. Okay, so microbial genetics will be the gene mapping techniques that we will be using for mapping the genes on the bacterial chromosome. Okay, so we'll be taking some specific, some typical part C kind of questions from microbial genetics that has been asked in the previous year question papers. And along with that, okay, at the end of the sessions, I request all of you to stay till the end, because at the end of the session, our team will share some link for some freebies, okay, complete free things will be given to you, some documents, some slides, okay, some presentation will be given to you, which are going to help you in your CSIR preparation. ठीक है, तो मैं सबको रिक्वेस्ट करती हूँ कि प्लीज स्टे टिल द एंड सो दैट यू कैन गेट द लिंक फॉर द freebies. And also, okay, as the regular joinees will know that we also conduct free Zoom class at the end of the session. Okay, so if you stay till the end, we will be having free Zoom session where you can clear your one-on-one doubt from any microbial question, genetics question with me. Okay, so that we will have after the session. So again, the team will share the link. You can join that free Zoom session link and we can have a question discussion. Okay, so because in this free session we will be discussing questions, but it might not be possible for me to answer and clear each and everybody's doubt. So I request that all of you note down your doubt. Okay, आप हर क्वेश्चन पे अपना डाउट लिख लीजिए, so that at the end जब हम एक जूम सेशन करेंगे तो उसमें सारा क्लियर हो जाए. ठीक है, तब आप अपना स्पेसिफिक डाउट मुझसे पूछ सकते हैं और फिर हम क्लियर आउट कर सकते हैं. Okay, right? So let's get started, everyone. Priyadarshini, Sachin, SV Creations. Yeah, good evening to you all. And let's start. Okay, so I'll turn off the video so that we can have an uninterrupted audio. Yeah, good evening, Garima. Okay, so let's start the session with relatively easy question here. Okay, where we are going to talk about, and this is a question from microbial genetics only, but here the question can be asked in part B also. Okay, since it is important for developing the concept, that's why I took it, so that we can start with some easy questions today and see. So this question is talking about, so DNA from a strain of bacteria with genotype A plus, B plus, C plus, D plus, E plus was isolated and used to transform a strain of bacteria that was A minus, B minus, C minus, D minus, and E minus. The transformed cells were tested for the presence of donated genes. The following genes are found to be co-transduced. Okay, A plus, D plus, B plus, E plus, C plus, D plus, and C plus, E plus. So ये वाला इंफॉर्मेशन जो दिया हुआ है उसको यूज करके यू हैव टू फाइंड आउट द ऑर्डर ऑफ द जीन्स ऑन द बैक्टीरियल क्रोमोसोम. Okay, so for that it is important to understand first of all what is transduction. Transduction is the transfer of genetic material from one bacterial cell to another via viruses. Okay, so so for transduction it will be delivery of genetic material between two bacterial cells. Okay, but we need a delivery agent here. Okay, and who will be the delivery agent? That will be the viruses or the bacteriophages. Okay, now when we use the word co means together. Okay, so for together, so when I'm saying transduce, that means a gene was transferred from the donor bacteria. There is a donor bacteria and a recipient bacteria. Okay, so a gene was transferred from the donor bacteria to the recipient bacteria via the delivery agent virus. Okay, that is what is transduction. Now, when I'm using co-transduction, means two or more genes are transferred together. Okay, तो together transfer करना है तो definitely एक gene तो transfer हो नहीं सकता है. So एक से ज़्यादा. Okay, it might be 2 or 3. Okay, like that. So this co-transduction will mean that two or more genes are transferred together from the donor bacteria to the recipient bacteria. ठीक है, तो अभी क्या जीनोटाइप दिया हुआ है? The donor bacteria's genotype is A plus, B plus, C plus, D plus, E plus, and the recipient is minus for all. ठीक है, now co-transduction कब हो सकता है? When the genes are present close together on the DNA. Yes or no? Suppose if you have this DNA, okay, and let's say 1, 2, 3 and 4. These are the 4 different genes present. Okay, so definitely 2 and 3, the chance of co-transduction of 2 and 3 will be higher compared to the co-transduction of 1 and 4. Okay, why? The simple logic of linked genes, that the genes which are present close together on the DNA, they have higher tendency of being linked, right? So, and if they are linked, then only they will be co-transferred. Okay, so here the transfer method is transduction, so the genes will be co-transduced. Okay, so the genes which are co-transduced is basically giving us the idea that they are the one which are present in strong linkage. Okay, and genes will be present in strong linkage only with its neighboring gene. Okay, so if A and D are co-transduced, means A and D are present pretty close to each other, right? Now B and E are co-transduced, means B and E are also present pretty close. Okay, now another one is C and D is co-transduced. Okay, so if C and D, so if you see A, so this D is getting co-transduced with A as well as D is getting co-transduced with C. What does that mean? That D has to be present between A and C. ठीक है, सो अगर D दोनों के साथ ही जाता है तो D can be with C also if D is present in the middle of A and C. Okay, and D can go with A also. Okay, now next one, what do we see? That C and E co-transduce. Okay, so the same way that we saw for D, so this time you see for E. So E is co-transducing with B as well as E is co-transducing with C. ठीक है, इसका सीधा मतलब क्या होगा कि E जो है वो B और C के बीच में प्रेजेंट है, राइट? सो E जो है B और C के बीच में है, सो E यहां पे नहीं होगा, E होगा यहां पे. Okay, so that C and E can co-transduce and B and E can co-transduce. All right, so the order we will get is A, D, C, E, B. So answer number 2, option number 2. Yes, Pooja, Arun, Priyadarshini, absolutely correct, everyone. Okay, so this is right, it's quite easy, isn't it? But the thing is that this type of question has been asked multiple times. Okay, so it has been asked previously also, and in fact in the last CSIR exam, the very last CSIR exam that happened in June 2022. In that also a question of that this was asked. Moving on to the next question. So a cross was made between HFR Met plus, ARG plus, Lu plus, STRS, okay, that is sensitive to streptomycin, with F minus strain, that is the genotype is given as Met minus, ARG minus, Lu minus, in which Lu plus X conjugates are selected. If the linear organization of the genes are Lu plus, ARG plus, Met plus, which one of the following genotype is expected to occur in the lowest frequency? Okay, so for this, it is important that you understand the concept of HFR cells. Okay, so what are HFR cells? So HFR cells are the one where the F plasmid has integrated with the bacteria's own genome, right? So when the F plasmid, when that particular HFR cell undergoes conjugation with an F minus cell, so it will the F plasmid will start moving, okay, and because the origin of transfer is there, and as the F plasmid start moving, so it will take a part of the bacterial chromosome also to the recipient cell. Okay, so whenever there is conjugation between HFR with F minus, okay, तो इसमें डोनर कौन होगा, रेसिपिएंट कौन होगा? कोई बता सकता है जल्दी से, डोनर और रिसिपिएंट कौन होंगे? Absolutely correct, Akshata. HFR will be the donor. Okay, why? Because HFR has the F plasmid, even though it is integrated with the bacterial chromosome, but HFR has the F plasmid. So from there only the transfer will initiate. Okay, so HFR will always be the donor. Okay, and the DNA will be transferred in a unidirectional way from HFR to F minus cell, which will act as the recipient. Okay. Absolutely correct. Jaya, Priyadarshini, yes, all of you are correct. So HFR will be the donor. Okay. Now the HFR genotype is written here. So let's write it down. So the donor's genotype is Met plus, ARG plus, and Lu plus. Okay. The recipient's genotype is Met minus, ARG minus, and Lu minus. Okay. Now, X conjugates are selected. X conjugates means the cells which are separated after conjugation. Okay. Now, what is the scenario with the HFR cells? That the HFR cells will transfer the genetic material from donor to recipient till the physical contact is maintained, because conjugation is a direct transfer. It requires physical contact. So, if the physical contact breaks somehow, then the transfer will immediately stop. Okay, so whatever segment that has been passed from HFR to F minus cell, so that segment will find its homologous sequence in the F minus cell, and then those genes, if any has been transferred to the F minus cell, can be incorporated in the F minus's genome, okay, which results in the formation of the recombinants. Okay, now the question is, which one of the following genotype is expected to occur in lowest frequency? Okay, now here you have to correlate to our understanding of a 3-point test cross. याद है सबको 3-point test cross? So in 3-point test cross, what we get? In 3-point test cross, which one has the least frequency? The least frequency is given by the double crossovers, right? The DCOs, the double crossovers. Okay. Now, why double crossovers? Because क्या होता है कि उतने छोटे से एरिया में दो क्रॉसिंग ओवर होना पड़ता है to get this double crossover product. Okay. So basically, more are the number of crossovers involved, lesser will be the frequency of that. Are you guys getting me? So more is the number of crossovers required, lesser will be the frequency of that particular recombinant. Okay. So this is donor, right? And this is recipient. Okay. So the maximum number of crosses that it can involve will be what? 1 here, 2 here, 3 here, and 4 here. Okay. Now with this many crossovers, that is 4 different crossing over points, what will be the genotype of the recipient? Okay. So recipient, what happens? This is the first crossover. So it is getting the MET plus. Okay. So from here it is getting, so I'll just circle it in blue. So from here it is getting MET plus. Okay. Then it is getting Arg minus. Then it is getting Lu plus. Okay. And then the bacterial chromosome, the recipient. Okay, so this is the particular genotype which requires maximum number of crossovers, and definitely the frequency of such a recombinant will be least. Okay, so what is the genotype? That will be MET plus, Arg minus, and Lu plus. Okay, so answer will be option D. I hope, Akshata, you understood. If you guys have any doubt, any confusion here, please write it down and we can discuss during the Zoom session. All right. So before we go ahead, guys, a quick announcement that the Shaurya batch for CSIR NET 2023 is starting from tomorrow itself. There will be coaching for CSIR NET December 2022 plus June 2023 as well as December. Okay, so June plus December is included here, and we also offer the AIM-NET and AIM-GET series also you will get. Along with that, you will get all completely free R&D internship. Okay, and one free access to all workshops conducted by Biotechnica. Okay, so that's a quick announcement for the Shaurya batch. If you guys want to enroll to the Shaurya batch, please quickly you can dial to the free number. Okay, the toll-free number mentioned here, and you will get the proper assistance. Okay, we even have scholarships. All right, so if you are looking for some scholarships, that also can be provided. So please contact us, and if you know somebody who wants to get the CSIR NET coaching classes from Biotechnica, please let them know as well. All right, moving on to the next question. So the co-transduction experiment was performed to decipher the linear order of 4 genes, A, B, C, and D. 3 sets of experiments were done, where transductants were selected for A gene in set 1, B in set 2, and C in set 3, and screened for co-transduction of other markers. Okay. So here this set 1 is selected for A. Okay. Here it is selected for B. This set 3 is selected for C. Okay. Now selected means what? That for sure you are checking whether that particular gene has transferred to the recombinant or not. Okay. Along with that, whatever gene has come, that is mentioned here. Okay. So basically in this set, you have 31 cells where A and B both were co-transduced. You have 3 recombinant cells where A and C have co-transduced, and you have 89 cells where A and D have co-transduced. Okay. Now, from this understanding and the discussion that we had about co-transduction, so which gene A will be most linked with? Can anyone tell me quickly? From this understanding, can you tell me quickly with which gene A is most linked with? Most linked will also mean that the distance is least between A and that particular gene. Yes, anyone? Which gene A will be most linked with? Yes, absolutely correct. No, Priyadarshini. Akshata, Pooja, absolutely correct. D. Because the number of recombinant is more in that, means those number of cells have got A and D both together, because A and D have co-transduced. When it can happen? When A and D are present close to each other. Okay, that means from here what we can find out, then A and D are close to each other. Okay, and see, the number of A and C recombinants is least. Okay, means A and C will be far from each other. Okay, that much inference we can make. Now let's go to set 2. So for set 2, we have what is the frequency, which is the maximum frequency is 78, and that is for B and C. Means B is most closely linked with C. Right? So we have AD most closely linked and we have BC most closely linked. Okay. Now next, what do we have in set 3? So for set 3, what do we see? That C is most closely linked to B. Okay, so this is B and C that we have found here also. And one important thing that we get is C and A. The frequency is 0. Okay, means what? That C and A, and also from this set 1 also we see that C and A, the number of cells getting C and A both is very less. Here तो in set 3 it is 0 only. Okay, so इससे क्या साबित होता है कि A और C for sure are the farthest two genes. Okay, and what else do we see? So for B can transduce with C as well as it can transduce with D. Okay, and the co-transduction frequency of BD is higher than BA. Right? Now this co-transduction frequency is basically, this co-transduction frequency will be inversely proportional to the distance. Okay, so bigger is the distance between the genes, lesser will be the co-transduction frequency, right? So co-transduction frequency is inversely proportional to the distance between the genes. Okay, so bigger is the distance between the genes, lesser will be the co-transduction frequency. Okay, now since here we see that BA co-transduction frequency is lesser than BD, that means distance between AB will be more than BD. Okay, so this criteria can also be followed in this combination. Okay, so A and C are farthest, and because of that co-transduction frequency between A and C we are finding least in all the three sets. Okay, B can co-transduce with D as well as it can co-transduce with C, right? And B can also co-transduce with A, but the co-transduction frequency with A is lesser than that between B and D. Okay, so if we take all the information here and correlate it with the order, so ADBC is the most probable order of the three markers on the gene. Absolutely correct, everyone. Answer will be option 4. Okay, next question. Now, remember everybody that all of these are previous year CSIR questions only. Okay, in fact, in the last session, lot of questions were asked. Okay, 2 questions came in part C from this microbial genetics itself. Okay, so 5 bacterial markers were followed for co-transduction experiment. The following table documents the observation of this experiment. Plus denotes co-transduction and minus denotes lack thereof. Okay, ND stands for not determined. Lack thereof means co-transduction did not happen. Okay, so here if you see this table is given, right? So this cuts are there. So cuts are there in those situations where it is same 2 markers. So leucine with leucine you cannot see, right? Arginine with arginine you cannot see. All right, so that's why the cuts are here. Okay, STR with STR you cannot see. Okay, now wherever co-transduction was seen, it is marked as plus. Wherever co-transduction was absent, it is marked as minus. ND stands for not determined. Now pick the correct order in which genes are arranged on the bacterial chromosome. Okay, so as you can see the trend that this kind of question has been asked multiple times, where they give you the co-transduction frequency either in the form of this kind of table showing presence or absent, or in, or how it was given in the previous question where number was given. ठीक है, तो दोनों ही सिनेरियो में या तो यहाँ पे टिक करके बताया गया है कि को ट्रांजैक्शन है नहीं है, या फिर कोई नंबर दिया गया है. ठीक है, तो उन दोनों ही सिनेरियो में हमको ये डेटा को एनालाइज करना है, एंड वी हैव टू गो बैक टू द सेम अंडरस्टैंडिंग दैट को ट्रांजैक्शन फ्रीक्वेंसी इज इनवर्सली प्रोपोर्शनल टू द डिस्टेंस बिटवीन द जीन्स. जितना जीन्स के बीच में डिस्टेंस ज्यादा होगा उतना ही कम चांस होगा फॉर देम टू गेटिंग को ट्रांसड्यूस टुगेदर. Okay, and lesser is the distance between the genes, higher will be the co-transduction frequency. तो यहाँ पे अगर देखेंगे तो gal and arch is co-transducing, right? Gal and arch, there is plus, okay, and gal and str, so gal and arch is co-transducing, as well as str and gal are co-transducing. ठीक है, अब इन दोनों सिनेरियो में अगर हम देखते हैं तो कौन सा मार्कर कॉमन है? गैल. ओके, तो इसका मतलब क्या होता है कि गैल विल बी प्रेजेंट बिटवीन आर्च एंड एसटीआर. ओके, अगर गैल दोनों के बीच में प्रेजेंट होगा, अगर ये ऐसे प्रेजेंट होगा, तभी तो वो आर्च के साथ भी को ट्रांसड्यूस कर सकता है और एसटीआर के साथ भी को ट्रांसड्यूस कर सकता है. ठीक है, सो गैल हैज टू बी प्रेजेंट बिटवीन आर्च एंड एसटीआर. Now next one, if we see LEU, LEU is also co-transducing with STR and MET. Okay, so if LEU is co-transducing with STR and MET, so similarly क्या conclusion निकलता है कि STR or LEU can co-transduce, and what LEU and MET can co-transduce. Okay, तो again इन दोनों में से common है LEU, that means LEU has to be present between STR and MET. Okay, तो अगर हमने STR को यहाँ पे place किया है तो let's see if this one works or not. Okay, so this is like a trial and error. Okay, so you have to analyze the result, formulate your combination, and then recheck whether all the findings that is given to you in the table or in any format is matching or not. ठीक है, तो हम फिलहाल ये लिखते हैं कि LEU is between STR and MET. Okay, and STR, if you see, is also co-transducing with LEU. Okay, so gal and arch can co-transduce, gal and STR can co-transduce, STR and LEU can co-transduce, and LEU and MET can co-transduce. Okay, what does that mean? That this order that we have written is possible. Okay, so out of all the options, check if this order is given or not. So you'll see that yes, this order is given: arch, gal, STR, LEU, MET. Okay, now again I will like to tell you or remind you that in this kind of questions, it is the genetic order that we are getting. Okay, it is not physical mapping, it is genetic mapping. Okay, so genetic mapping means it's a relative mapping. All right, so when we are talking about relative mapping, अगर आपने ये वाला order लिखा है और order उल्टा दिया हुआ है, means MET से शुरू करके फिर दिया हुआ है MET, LEU, STR, gal, arch. ओके, तो अगर ये वाला ऑर्डर भी दिया हुआ है, देन ऑल्सो दिस इज करेक्ट, बिकॉज द रिलेटिव पोजिशनिंग ऑफ द मार्कर्स आर इन अकॉर्डेंस विद द फाइंडिंग. ओके, सो आपने जो भी ऑर्डर निकाला है, जरूरी नहीं है कि वो दिया हुआ, उसका उल्टा भी दे सकते हैं. ठीक है, पर अगर उल्टा दिया हुआ है तो भी वो करेक्ट आंसर होगा. तो इन केस आपने जिस फॉर्मेट में लिखा है, यू चेक इट एंड यू डोंट गेट एन ऑप्शन फॉर दैट, तो उसको उल्टा करके भी चेक करो. Probably in that order it will be given. Okay, अगर आपका एनालिसिस सही है तो either this or this will be given. Okay, because it is genetic mapping, so it is relative positioning of the markers, not absolute positioning. All right, तो ये आप ध्यान रखिएगा अगर कोई भी genetic mapping का क्वेश्चन आता है तो. All right, next question, let's have a look. So using the interrupted meeting, 4 HFR strains were analyzed for the sequence in which they transmitted a number of different genes to F- strains. Each HFR strain was found to transmit its gene in a unique order as summarized in the table. only the first 5 genes are scored. Okay. Which one of the following correctly represent the gene sequence in the original strain from which the HFR strain were derived, as well as the place of integration and polarity of the F plasmid? Okay. See, we have already talked about that what is HFR strain. So HFR strain will be the strain where the bacterial DNA is incorporated with the plasmid DNA. Okay, let's say blue is the plasmid DNA. What is this plasmid? This is the F plasmid. Okay, now this F plasmid will have the origin of transfer. Okay, and so depending on where this F plasmid is integrated and in which orientation the F plasmid is integrated, the genes will be transferred in a particular order from HFR to F minus cells. Okay, so ये जो different strains है HFR के, तो वो basically क्या है? वो सारे ही same cells के strain है. That means they all will have the same genes present at the same position. What is different in them is the position where the F plasmid is integrating with the bacterial chromosome and the orientation in which the F plasmid is integrating. ठीक है, सिर्फ यही दो difference होगा, otherwise everything will be same in this HFR strains. All right, so इसमें दिया हुआ है the order of transmission. So first ये जा रहा है. That means the order of transition for this HFR, for example, this HFR strain 1 will be A, then B, then D, then F, then H. Okay, so ये वाला order होगा strain 1 के लिए. Okay, now let's draw the diagram. So how do we do the gene map? So कोई भी अगर ये वाला situation दिया हुआ है, so this is an example of interrupted mating experiment. Okay, so we see the order in which the genes markers are transferred from HFR to F minus, right? So सबसे पहले क्या करेंगे कि we will make a circle because bacterial DNA is a circular DNA, right? Now for first strain, it is A is going first, then followed by B, D, F, H. ठीक है, तो एक के बाद एक लिख लेते हैं. A, then B, then D, then F, and then H. Okay. Now, अगर integration site की बात करेंगे of the F plasmid, so F plasmid is integrated in such a position and in such orientation that A is going first, followed by B, D, F, H. Okay. What does that mean? That the integration, the F plasmid must be at this position and present in this orientation. So arrowhead shows the direction of orientation. Okay, so that, so यहाँ पे अगर present होगा तभी A पहले जाएगा. So it is just like how it is pulling. Okay, so A will be pulled first, then B, D, F, H, and it can only happen if the circle here, the DNA here is moving in the anti-clockwise. Okay, so in the anti-clock direction. So this is how we have placed the first strain. Okay, now let's do the next one. So BALMK. So if you compare this two, so this AB is common. Okay, but now it is in the opposite direction, starting with B. So BA to aa gaya hai, means on the other side of A will be L, then M, then K. Okay, now यहाँ पे कहाँ present होगा? So it will be present at this position and in the opposite direction, like pointing towards D. Okay, then only अगर F plasmid यहाँ से निकलता है तो it can be BALMK. Okay, so likewise you can map the other genes also. So for strain 3, so this is for strain 2. So for strain 3 it is MK, JGH. So MK already has, so J, G and H also we have mapped. Okay, and M is again going first here, so there will be the next site of integration or for the third strain and pointing towards L. Okay, and for the fourth strain it is FHGJK. So where it is FHGJK. So F is going out first, so the orientation and the integration site of the F plasmid will be present at this position pointing towards D. Okay, so this is the map that we have created by analyzing the table here. Now you match it with the options. Okay, so this is option 1, 2, 3, and 4. So you will see that option 3 is matching with the map that we have created. Okay, so again guys, make sure that this is again relative positioning. अगर आप ABDF इस साइड से ना घुमा के अगर आप ऐसे प्लेस करते, ठीक है, तो क्या आपका मैप गलत हो जाता? नहीं. फिर से आपको देखना था कि रिलेटिव पोजीशनिंग कहां पे है, whether B is present between A and D or not, whether F is present between H and D or not. Okay, so ये relative positioning है. तो अगर आप ABDF इस साइड से प्लेस कर रहे हैं या फिर उल्टे साइड से प्लेस कर रहे हैं, डज नॉट मैटर. Okay, I hope all of you understood this. Okay, many of you have already given the correct answer. Yes, Akshata, Tithi, Arun, Priyadarshini, correct. All right, now moving on to another question, and this question was also asked in the previous year question paper. देखा है किसी ने? Last CSIR exam में ही ये question आया था. Did you guys check it out? थोड़ा जाना पहचाना लग रहा है क्वेश्चन देख के. लास्ट CSIR पेपर में ही था ये वाला क्वेश्चन. No, Akshata, so I think you have not checked the last CSIR paper. Okay, check it out. Okay, take the question paper. All right, और उसमें देखो ये वाला question है. Okay, now let's see what the question is talking about. Transduction was used to map FADL gene. Okay, the result of two-factor crosses between FADL and two linked markers per F and arrow C are shown below. Okay, so there are three marker genes. What are they all? FADL, per F, and arrow C. Okay, now you have donor and recipient. Okay, so you are selecting one marker, and what is the second marker in them that is mentioned, and number of such kind of cells that you have obtained is mentioned here. Okay, now first situation, what is given? So the data is given here. Okay, so what are the different recombinants you are getting, and what is the number of those cells is given in this column. Which of the following is the correct map of the three genes? All right, see, so यहाँ पे donor का genotype क्या दिया हुआ है? Let's write it down. FADL and per F plus. ठीक है, the recipient का genotype क्या दिया हुआ है? The recipient's genotype, so I'm writing it like this so that to make sure that both of them are on different chromosomes. Okay, so those two horizontal lines are showing different chromosomes. So here it will be FADL plus and per F. ओके, सो इसमें से डोनर कौन है? ये वाला डोनर है, ये नीचे वाला रिसिपिएंट है. ठीक है, तो अगर को ट्रांसडक्शन होगा, सो फर्स्ट ऑफ ऑल हाउ डू वी डिटरमाइन दैट वेदर द जीन्स आर प्रेजेंट क्लोज और नॉट? सो दैट वी विल डिटरमाइन विद द हेल्प ऑफ को ट्रांसडक्शन, अगेन विद द सेम identification that co-transduction frequency is inversely proportional. Co-transduction frequency is inversely proportional to the distance between genes. Okay, so co-transduction यहाँ पे कब होगा? Co-transduction will happen when both the genes from the donor is getting transferred to the recipient. ठीक है, तो ये वाला जो है who is per F plus and LDL minus, okay, which is the genotype of our donor here. Okay, so this is the one who is co-transduced or co-transductant. समझ में आ रहा है? So this is the variety which has been created after the co-transduction of genes from donor to recipient. ठीक है, तो अगर ये co-transductant है, so what will be the co-transduction frequency between FAD and per? So if I write it here, co-transduction frequency between FAD and per. So what it will be? 200 divided by total transductance, which is 1000, right? 800 plus 200 will be 1000. So which will be equal to 20%. Okay. Now, exactly in the same way, we have to analyze for the other sets as well. Okay. So in the other set, so for better representation, let me write it with two different colors, right? So जो donor है उसको मैं blue से लिख रही हूँ, that is FAD L, okay, and arrow C plus, okay, और जो recipient है उसको मैं blue से लिख रही हूँ, which is L plus, for short I'm writing, it is FAD L plus and C minus, okay, or donor. So blue is what? It is donor. रेड वाला जो है रिसिपिएंट है. सो अगर को ट्रांसडक्शन हो रहा है, दैट मींस the recombinant will be FAD L and arrow C plus. So FAD L and arrow C plus means ये जो 400 है, ये वाला नंबर है को ट्रांसडक्टेंट का. Okay, so again हमको ये मिल जाता है, तो we can calculate the co-transduction frequency between FAD and arrow C. Okay, so how much it will be? 400 divided by 1000 again, so it will be 40%. Okay, now once we do that, so again what we have to do? We have to calculate the for the next set, right? So we have to calculate for the next set. So what do we have here? So in the next set, we have got this one, arrow C plus and per. Okay, so here this is the donor and this is the recipient. So again, you have to look for the co-transductant. Co-transductant will be arrow C plus and per F, which is this 500. So this is co-transductant. Okay, so what will be co-transduction frequency? So I'll write it here down. So between arrow C and per F. So how much it will be? 500 divided by 1000. It will give us 50%. Okay. Now, once you have calculated the co-transduction frequencies, next we have to relate it with the distance. Okay, so here again we have to do the same correlation, that co-transduction frequency is inversely proportional to distance. Okay, so since co-transduction frequency between FAD and per is least, means FAD and per has to be present farthest. Okay, so for example, of FAD and per, it will be 20%. Co-transduction frequency of arrow and FAD, I will write it here, FAD and arrow. Okay, it is 40%. And co-transduction frequency of this much we have calculated. Now, how to correlate it with the distance? So co-transduction frequency is inversely proportional to the distance between the genes, right? So when we correlate it, okay, so if FAD and per co-transduction frequency is least, means they will be present at farthest distance. So if you have two genes, so what will be the farthest distance? Farthest distance will be the scenario where they are present at the ends. Okay, that means FAD here, per here. Okay, now what about arrow? Arrow has to be present between FAD and per. Okay, but more towards which? So it will be placed more towards per because co-transduction frequency here is higher. Means that the least distance is there between arrow and per. Okay, so arrow will be present here. Okay, so co-transduction frequency between arrow and per is 50%. Arrow and FAD is 40%. Okay, there's a slight increase in the distance, and FAD and per, which are highest distance from each other is 20%. Okay, so when you compare this with the maps that are given to us, so which one matches? So the one that is matches will be the okay. So option number 2. Okay, where FAD, I will just clear it all out. So answer will be, this is option 1, 2, 3, and 4. So option number 2 will be the answer. Where FAD and PER at the maximum distance, okay, 20% co-transaction between them. Arrow is present in the middle, okay, with, although in the map it is not showing correctly, but arrow will be present little closer to PER. Okay, why? Because the co-transaction frequency is higher here. Okay, and then the co-transaction frequency between arrow and PER is 50%, FAD and arrow is 40%. All right, everybody. So with this, I would like to wind up the live session here. I request the team to put the link for the freebies as well as the link for the Zoom classes. So the link is coming right here, guys. I expect all of you to get the benefit of it. Okay. So the link for the live classes, the Zoom class will also be given here. So please join the link. Okay. And the live class will start soon. Thank you everyone for joining. Thank you all the students for joining here. I am looking forward for you all to join the Zoom class where we can have one-on-one discussion on any doubts that you have. And if you don't have, we can also see some extra questions. Please come to Zoom class and we can do the further discussion. Thank you everyone. See you in the Zoom class soon. Again, I request the team, please send the link for the Zoom class